Good morning, everyone, and welcome. Welcome to this session. This is our first overview session for bioinformatics of infectious diseases. And in this session, what we want to do is review the program schedule, resources that we will use in the program, and speak in greater detail about the topics that we will cover throughout this program that will last starting from May 4th all the way until June. So we're going to get started here. The purpose of this program is to introduce you to some of the tools, some of the bioinformatics tools and data sets and project ideas, and essentially provide you with an opportunity to develop a meaningful project where you can apply some of the bioinformatics analysis methods to publicly available data sets and demonstrate your understanding of a specific topic based on data. So I want to start by just kind of clarifying bioinformatics for infectious diseases. Infectious diseases are disorders that are caused by organisms, other organisms such as bacteria, viruses, fungi, or parasites, as opposed to, let's say, chronic diseases that are related to changes in the body of the human or the host. And some infectious diseases can be passed from person to person, so that's why they're called infectious. They're transmitted by insects or other animals, and you may get others by consuming contaminated food or water, being exposed to organisms in the environment. So many infectious diseases are prevented by vaccines, and a lot of those cause an acute reaction, which are treated by antiviral drugs. So in the context of this program, what we want to do is speak about some of these concepts, introduce you to some of these pathogens. The pathogens are those organisms that cause disease, and these are both viruses and bacteria. We will not really talk about parasites and fungi, but we will focus a lot on viruses and also cover a little bit of bacterial infectious diseases. But we will do this in the context of bioinformatics. So we will really speak about methods of analysis for omics data, and we'll talk about how to analyze this data to get meaningful results from the analysis. I'm seeing that maybe if anyone is having any technical issues, again, please try to look at your settings, and we will have a recording later on of the session that you will be able to review. Okay, so first of all, about myself, my name is Ilya Brodsky. I'm the co-founder and CEO of Pine Biotech. Pine Biotech was established with a vision to enhance human health and well-being by enabling biological research and discovery with relevant data, software solutions, and support like training that we're doing now. Our mission is to simplify bioinformatics and advance research through our modular and intuitive multi-omics analysis platform powered by human experience and artificial intelligence. We offer training, research support, and big data analytics tools designed for research and biomedical discovery. And our mission is to support cutting-edge research and solve big data challenges by making our solutions easy to use and train those interested to use them to arrive at sound and reproducible results. I am not the only one that is organizing these programs, so I do want to say a couple of words about our team. I'm honored to have been working with people that have much more experience in this area than myself, including some of the other co-founders in the company, Dr. Alfred Tauber, who comes from a medical background and has deep expertise in oncology, immunology, and biochemistry of inflammation, who has a long-standing recognition for his generous support of many international projects and research activities. And one of these projects is the establishment of the Tauber Bioinformatics Research Center at the University of Haifa in Israel. And that center is essentially at the core of what we are doing. The director of that center is Dr. Leonid Brodsky, and he's the director starting from 2012. He's an expert in bioinformatics and biostatistics and has numerous publications on bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches to biomedical data analysis with a focus on big data. Some of these tools have been widely used in basic and translational research in biotechnology, pharmaceutical R&D, agricultural projects, and biomedical research. But what we will discuss today is also a result of hard work by some of the people that we have here on the left. Julia Panov, who is an expert bioinformatician and has helped prepare many of the materials that we will use today. Avi Titievsky, who is a computer scientist and leads the development of the T-Bioinfo platform that we will utilize throughout the program. Mohit Mazumdar, who is in charge of business development for Pine Biotech. Joshua McKendall, who maintains the educational web portal that we will leverage, and Bipsha Biswas, who is our online community manager and support specialist, who I'm sure that you have had the chance to interact with already. So just to make sure that everybody is aware with our great team, familiar with our great team. So let's start talking about this program. The program is designed to provide you with some of the tools and data sets and introduce you to some of the concepts related to bioinformatics of infectious diseases. It will last from May 4th to May 28th. In reality, after that, you will have more time to start working on your own independent project. And that is the important component of this program, is not only to provide you with information, but also enable you and support you in the design and the development of a project of your own, that you can try and apply these tools to a data set that you have selected, and you understand what might be a meaningful data-driven project where bioinformatics can play a critical and important role. So let's talk about first the schedule of the program and some of the resources that we will use. And I'll talk a little bit more about how to register if you have not registered already for all the different aspects that we are combining into this program. So bioinformatics for infectious diseases is really focused on three things. We will try to understand these pathogens and the diseases that they cause. So we'll talk about the different animal reservoirs, hosts, and vectors of disease. We'll talk about viral evolution and replication. And we'll talk about also bacterial infections. So we need to understand those pathogens. We'll talk about some of the analytical approaches, how to detect viral genomes in next generation sequencing data, how to detect presence of non-host genomes in next generation sequencing data in general. How do we understand the specificity of our analysis? So once we determine some kind of a presence of a known organism, how do we know what we have found, how similar is it to some of the more known pathogens? We'll talk also about vaccine design, antivirals, and antibiotics. So how does the treatment fit into that understanding? And then also we'll talk about epidemiological trends and drug resistance. So epidemiological trends might include transmission. So how do we look at transmission from a bioinformatics standpoint? How do we look at drug resistance or efficacy of different intervention measures from a bioinformatics standpoint? We'll also talk about, not in general terms, but we'll take curated projects and data sets. And we'll actually, we have prepared a number of these that I'll cover in a little greater detail throughout this session. But they include epidemic outbreaks that have generated a lot of data, like Ebola, Zika, dengue. We'll obviously look at coronaviruses like SARS, MERS, and COVID-19. And we also prepared a number of materials for Mycobacterium tuberculosis and leprosy. So we'll talk about some of these. What's interesting is that there is an exponential growth of some of these resources, including publicly available data sets. We, in fact, published a couple of blog posts recently that talk about some of the genomics resources for virology research and education that you can look on our website right here. We also try to estimate the rate of submission of genomic data related to viruses. And what we see is, at least in the recent outbreaks, including SARS, Ebola, MERS, Zika, and the current COVID-19, we see a tremendous increase in the availability of this genomic data that provides us with a very clear view of how the pathogen, the source of this pandemic, evolves and spreads. And so we can really understand things from a data-driven perspective in a very rapid way. Of course, what's needed for that is the experience and the understanding of the data so that we can understand where the data is coming from and how to analyze this data with a good sense of where to look for, what kinds of patterns and what kinds of specific changes from whatever we are comparing this to. So I think that training is a key aspect of being able to understand how bioinformatics fits into this. To be able to speak about this, as I mentioned, we prepared a number of resources that we will make available throughout this program to the participants of the program. And these are essentially projects based on public domain data sets and based on some of the materials that you would find in a publication. So we actually reproduce some of the aspects of a publication's analysis that the authors have submitted. We take elements from that data set. Okay, I see that some people are having issues with audio. And then if anyone is not being able to hear, Deeksha, maybe can you let me know if you can hear me?
Yes, Celia, I can hear you clearly.
Okay, great. Okay, well, sorry for those that are not able to hear. Excellent, thank you, Ryan. So we prepared some of these materials. I think they provide an in-depth and well-characterized data sets that we can leverage to really understand where to look in some of these repositories and how might we adapt some of those approaches that we see to a challenge that we have identified. So we have here a project on tuberculosis, a ultra deep sequencing called circular sequencing or CIRSEQ of polioviruses. We will look at enterovirus D68, which causes acute flaccid myelitis in kids, and it has been a big public health concern recently. We'll take a look at the outbreak of Ebola virus in West Africa in 2014, 2015. And as you know, this is something that is coming back and has recently emerged again. So a very important aspect of infectious diseases. And of course, we will dedicate a significant time to talking about COVID-19 in the context of coronaviruses in general. And we do have a lot of these materials that you can see online to just kind of understand what that means. And here with this data set, we will actually go much deeper and talk about in greater detail in a more conclusive way how we can analyze this data and what can we understand about this important pathogen. So let me clarify some of the registration that might be a little bit confusing for some of you. So Bibsha here in chat posted a link to this landing page where you can pre-register. So we ask you to just fill out a form on that page. And those details come to us and we try to evaluate that you are a good fit for this program. So obviously this program has a certain level that we expect you to be familiar with some molecular biology terms, with some data analysis terms. We feel that the materials are user-friendly and well adapted to a beginner. But what we have seen is that some people at maybe undergraduate or even school level are interested in joining some of these programs, and we have to make sure that they do have sufficient knowledge to be able to make sufficient progress during the course of this program. So we do review some of your submissions and we reach out by email. And after we reach out by email, what you will probably get is a link that will ask you to register on our website. And on the website, it's a free registration. So you have to create a username, and then you are registered and you have an account. And you can use that account to access some of the freely available resources that we provide there. But also you can then continue to register for the full program. And so in the full program, what you will get is an email. And this email will include the program curriculum and also a link to the registration right there. So when you're looking to complete your registration, you want to access all of the sessions of the program, make sure that you click there. And when you click there, what you will be getting is essentially a program license. The program license, I'll speak about what it includes in just a moment, but the purchase happens here. We do have a discount code, viral30, that you can use to get a 30% discount from the total price. And this price actually goes to support some of the activities that I will talk in the next few minutes. Okay, so Bibsha, if you could again, just please post a link to the registration page and to the checkout page. So if anyone has any questions, they can take a look at those links throughout the session, and then we can answer any questions at the end. Okay. Okay, so what does this license include? This license provides you with access to some of the materials that will be available for the program participants. So you will be able to leverage those materials to learn about some of the basic analysis methods. So for example, how do we analyze data? What statistical terms, what molecular biology terms do I need to know to be able to go through some of this material that we're going to be doing in sessions, and also provides you with a tool. So if you're a bioinformatician, if you know how these tools work, you will be able to use those tools however you like to. But the data sets that we will use are large. It's unfeasible to be able to analyze those quickly on your home computer, on a laptop. And so you will be able to access our platform, which is designed for big data analysis, and use that platform to analyze the data that we discuss in the class. Also providing us with a way to monitor what's going on and provide you with any kind of feedback that is needed for you to be able to successfully complete those pipelines. So these things are separated onto two different links: server tbioinfo and edu tbioinfo. That's where the educational materials are hosted. This is the analytical platform, and they are interconnected. So we will talk about this in a second. There's a question here that the membership expired. So the membership actually starts on May 4th. As some people registered before, you might have gotten some email notifications about your membership expiring. However, you do have access for 2 months. So it's going to be starting in May and all the way till the end of June. And the purpose is to have access to these materials, the data sets that we have curated and designed a step-by-step tutorial on how to reproduce some of the analysis that the authors did, but also access to the platform. And that access is a separate login. So make sure that you add Bibsha, which is marketing@pine.bio, to your contact list, because she will be sending you your access information to the platform. Okay, I will pause for questions. If anyone has any other specific questions, feel free to add that into the chat, but we will pause for questions throughout this session. So what does the platform provide us? What kinds of tools are we going to discuss in the context of this program? So one important aspect of understanding the process of infectious diseases is being able to analyze gene and isoform expression using RNA-seq. So we will be able to look at the expression of genes in response to viral infection or bacterial infection. We'll be able to understand the immune response. We'll be able to map the specific gene and isoform level information onto pathway and gene ontology level of information. But more than that, we'll be also able to use data mining and network analysis and machine learning to be able to find those signals that are going to be integrating some of the data that we find in these large data sets. So we'll be looking at that section as well. We'll be looking at, for example, a project that you see right here, and I'll just play this little demo. This is how the platform looks and works. So this is an example project of response of different types of cell lines to different genomes with modified insertion or deletion of a certain gene. So you can see here that ORF6, which is one of the polyprotein in SARS, and SARS-2 produces the ORF6 gene. And you can see here how over the course of time, post-infection, the cells respond in very different ways, right? So you can see here mock infection, wild type is the blue, and the green is the ORF6 essentially mutant. So this type of analysis helps us understand how does a certain gene play a role in the response of a host cell to the infection by a certain strain of a virus. So we'll talk about some of these examples. Now we'll also leverage mutation variant analysis, and we'll look at a lot of different genomes that have been made publicly available. But when we have a lot of data, one of the questions is going to be, how do we actually analyze this kind of quantity of data, and how do we make sense of the most significant mutations that can explain differences between different groups of data sets? So we'll talk about some of that data set, and we'll talk about how we can make a meaningful analysis with the data that we have. And what you see is a pipeline that will help prepare the data, structure the data, so that we can analyze it for significant mutations. Now, what do I mean by significant mutations? Here's an example, right? So once we found some of those significant mutations, we need to understand what impact will it have on the function of a specific protein that is produced, for example, by a gene encoded in that genomic sequence. So we'll talk a lot about different proteins, organization of the genome into coding genes or protein coding genes, the function of those proteins, the interaction between those proteins, and how do we understand the significance of mutations that are going to be important for us to interpret the data that we have. We'll also talk about evolutionary study and association. So we'll, for example, understand how different genomes are related to each other, or how different genomic variants are associated with a phenotype. For example, in drug resistance, when we talk about tuberculosis, how can we select those specific mutations and interpret them from that standpoint? And another approach here is multi-omics integration. So if we do have data on cellular response or host response, how do we then understand how the different variants and changes in the genome can be associated with that kind of, again, omics data, right? So that's where the key multi-omics analysis comes from. Now we have a number of webinars already on some of the elements of this kind of analysis, but just to illustrate, we will have a number of comparisons between, for example, the novel coronavirus and previous coronavirus outbreaks. We'll learn how to perform such analysis, but we'll also be able to link the genomic data to data at the protein level. So what you see here is a tool that we designed that takes in the output from the platform that we just went through, and then helps us understand the position of different mutations and how can we interpret those mutations in the context of the proteins that those specific regions make. So we'll use some of that. We'll also use the same tool to understand the function of vectors. So those are insects, for example, mosquitoes, that carry the virus and spread the virus. And of course, the virus is under different evolutionary pressures as it is inside the host, for example, a human, or it is transmitted by a mosquito. And so we'll take a look at how analysis of data between those different hosts can help us understand the response of the virus to some of those evolutionary pressures. Beyond that, the platform offers additional tools that we will not, unfortunately, have the time to cover in the program, in the training program, but I did want to mention that it offers mathematical modeling of viral replication and the process of infection. So once we generate the right kind of data, we really can describe that data in biological terms. How do we translate that understanding, biological understanding, into mathematical terms and use ordinary differential equations to prepare a model and then be able to simulate that model to evaluate the different types of relationships that we have between factors affecting that infection and spread of the virus. We will also talk about some of the other tools that are available. A big part of generating the right type of data is being able to analyze microscopy images. So we do have a section dedicated to that. And then obviously screening and docking of 3D structures is a topic that a lot of people are thinking about in the context of in silico drug design or even the understanding of vaccine design. So we'll talk a little bit about 3D structures, and we'll talk about some of the strategies for that kind of thinking. We won't really do a lot of hands-on because we will be focused a lot on the omics data first. So again, as Beepsha mentioned here, SecNav is a tool that we developed to be able to visualize a number of outputs from the platform, and you're welcome to explore. And as we go through the program, we will enrich this specific demo version of the SecNav so that you can explore and interactively understand how the data looks. So just briefly, I will go there. So this is the platform. The platform is going to be available for all the participants, the paid participants of the program. If you're not a paid participant of the program, but you're interested in a demo of this platform, we can create a demo account for you and you can play around with some of the demo pipelines. But we will also discuss how to then apply some of these tools to real projects that are research-based. And so we do offer research licenses for that. And just to illustrate how that works, let's quickly take a look at how we have, what you can learn from using this pipeline. So again, a lot of the methods are publicly available, and a lot of the data sets that we will analyze are also, actually all of the data sets that we will use are publicly available data sets. So we want to be able to reproduce results that have been published and discuss those in terms of the data and the analysis that we're able to do ourselves. So this is an example from Nature Medicine that was recently published in March that describes the origins of SARS COVID-19 or SARS-2. And here you see a phylogenetic analysis of different host genomes and the comparison by looking in specific regions of that genome. So we'll try to reproduce a similar analysis where we essentially take those genomic sequences, understand how they can be aligned to each other, so multiple sequence alignment. We'll talk about the relationship between genomic data at the nucleotide level and amino acid level, and how those have specific physicochemical properties that we need to understand and interpret their function. But also in terms of evolution, we'll talk about the distance between different codon changes. And so when we talk about that transition from nucleotide level to amino acid level, what we need to understand is that those changes are not as simple from an evolutionary standpoint from one to another. So we will talk about how to incorporate that into our phylogenetic analysis. We will also talk about different phylogenetic tree building and models. And so we'll talk about, for example, what is the basic concept of this evolutionary analysis, and how do we select the right methodology for the analysis that you're interested to do. And then we will talk about the results. So I'll just briefly show you some of the outputs and why it's important to talk about the different results that we see here. So including visual results, including results that are in some kind of a file format that we can then take to a table manipulation software or some of the other tools that you might already have. A question here, how can I get a demo account? Beepsha, please, you can share that information with README. And I'm just going to also demonstrate the sequence navigator. So once you get that output from the T-Bioinfo platform, how can you actually visualize that data? So this is just an example here where we are looking at, for example, different strains of coronaviruses, and we're looking at their change from a reference genome. And what's important here to understand is not only accumulation of those mutations, these are bins across the genome, but also the impact on the different proteins that those sections encode for, and especially the structural component of those different proteins. So a question from Ryan here. Does the platform during pre-processing de novo support viral classification and filtering for depletion of numerous mRNA, miTRNA, and non-coding RNA, etc.? So we will talk about the process of data preparation. So how is viral metagenomic sequencing, what does it mean, and what exactly happens during that library preparation step? And then how does that apply to the analysis? So we will talk about that. In fact, our next session is going to be on next generation sequencing, and we will talk about those different strategies for understanding how the data was prepared and what are we expecting to find, including some of those other non-coding RNAs and other information. Okay, so again, you're welcome to play around with that, and Beepsha can help you set up a demo account if you're interested. Okay, so we did get to questions. We already answered a couple of questions, but I will pause here for just a few moments to make sure that if anyone else has any questions about what we talked for the few minutes in the beginning, I'll be happy to answer that right now. So we talked about what the program will cover in terms of what resources and what tools. And next, I will go into the schedule, and we'll talk a little bit about the schedule. We talked about the process of registration, and you have the links if you have not completed the registration, and also the different things that you will get access to. Do we need minimal computer requirements to perform the analysis? So again, all of the analysis that we will be doing together will be done on the platform. I will talk a little bit about how the platform is connected to the educational resources, in other words, to the projects, the data sets, etc. But you do not need any kind of computational resources on your own. So you can access this from a Chromebook. The only thing that you do need to have is an ability to store and upload data sets. These are going to be tables in Excel. So if you're able to open files in Excel, if you have an ability to download those to your computer or somewhere, that's all you would need. Okay, how many hours a week will the program require from our parts in terms of effort on average? That's a very good question. What I would say is let's answer this question after we go through the schedule, which is what we will do next, okay? But just in general, I would say that a lot of this is up to you. So we hope that you can leverage this program to develop a project of your own and be able to complete the project on your own. That's why the project, the sessions of the program are scheduled for only 1 month. And then at the end, you're given another month to fully dedicate that time to working on a problem of your own and hopefully get to a result. If you're not able to do that or don't want to do that, you will still benefit from doing the online materials and participating in the online sessions. And yes, there will be certificates given at the end, so we'll talk about that in just a second. Okay, any other questions? So video lectures, yes. We will have video lectures. They're going to be interactive lectures like this. Today we have participants that are also not a part of the program yet. And so after we have our group that everybody's registered, what we will do is we will review progress of participants in each session, address any questions or challenges that they might have faced performing the analysis or going through the materials that we have shared. We will have an interactive Q&A session just to make sure that everybody understands the material. But a lot of this is also going to be asynchronous. In other words, that the materials could be completed on your own. And we have some of those materials available, and I'll actually show you a few of them in a couple of minutes. And we will record the sessions, and then we will send you that recording to all the participants. And the cost is $150 before the discount. The discount is going to end when we start the program, which is going to be Monday. So make sure that if you want to still register today or over the weekend. Again, any questions about discounts, payments, how to register, etc., we'll be happy to answer that. Beepsha will be able to handle that. And if you have already enough bioinformatics background or are interested to apply the tools to a research question, we're happy to support that as well. Okay, are there any other questions, or should we move on? Okay, how can I make the payment? Again, we will send you the information, but we have some of the links that you can start from pre-registration and then all the way to being a part of the membership. So Beepsha, please share that. Yes, the sessions that are going to happen, and let me go through the sessions next, and you will see every one of those sessions does have a time. If you're not able to join during that time, you will get a recording. Okay, and Mikhail, considering the previous experience of participation and the fact that your projects are very well prepared, how many projects can be taken as part of this course during this course? As many as you wish. As you know, we work in parallel with the programs to make sure that materials are somewhat structured, and we hope that you will be able to navigate those. But of course, we're able to answer any questions that you might have as you go through the projects as well. We estimate that a project takes at least a few days to complete the way that it was prepared, but they're designed to have an open ending. So you don't have to just replicate the analysis that we did. We hope that you will try and do something additional with a new data set or modifying the analysis itself. So it's really up to you. We only provide this for 2 months. Yes, again, yes, in terms of registration, the registration will be expiring at the end of June. Yes, so from May 4th until the end of June, June 30th, you will have access to materials and the platform, and the materials that you registered for throughout the session, you will be able to complete throughout that time. Okay, just to be clear in the program, will we have access to the platform that by enrollment we will receive credentials for the duration? Exactly, yes. So for the duration of the program, you will have access to the platform, and those credentials will be active, meaning that you will be able to analyze public domain data, and specifically the data that we provide or data that you find and submit as a project. Are all the small courses that are within the EDU web free? Some of them are free. Some of them have minimal prices like $10. And so, yes, you are welcome. If you're not familiar with bioinformatics, I would recommend starting with Introduction to Bioinformatics. If you are familiar with bioinformatics, then I recommend taking the more advanced courses. Not all the courses are geared towards infectious diseases, but they could be useful. And we'll talk about that in the next few slides. And if you're registered for the program, by the way, you have access to all of those courses that we will discuss here in the next few minutes. Okay, so let me go on so that we can make sure that we cover the schedule of the program. So if once you're registered, you will be able to, in fact, let me go there. You will be able to see the program and what is included. Okay, so once you are registered, you will see some of the sessions that we will discuss next um, here. And you can register for individual sessions if you're not interested in the whole program, or you can register for the full program as well. So you can navigate like this and see some of the coming up sessions, and you can see that they will end up with all the way into your independent project. So let me also show you there's a syllabus right here. Maybe it's easier to see the overview like that and some description of each one of these sessions. And I'll talk in detail about these sessions here in a few minutes. So again, we will start on Monday, May 4th. And then you can see throughout the sessions what topics are actually going to be covered. So please take a look at this so you can see what we plan to cover. So now let me go over this together with you and answer any questions that you might have about that. So we will start next session is going to be on next generation sequencing. So as you know, and as we already discussed, these original sequences of viruses come from a host. And the host obviously has their own genomic information in them, and not all of the viruses or bacteria inside might be harmful. So we'll talk a little bit about what next generation sequencing data looks like, and how do we extract from there relevant information to study an infection, right? So when we have an infection, how do we first find specific reads that represent that infection that we're looking for? And we'll talk about different strategies for doing that, how we have some library preparation techniques that can help us amplify specific types of genomes, or we'll talk about how we just look for everything and then from everything that we have available, extract whatever we need, and what are some of the analysis methods to do that. The second session is going to be dedicated to multiple sequence alignment. So once we have identified some presence of some specific genome, how do we focus on specificity and understand relationship of what we have identified to other more characterized and known genomes that we can understand by comparison how they are related, whether they come from the same origin, and how are they different. And then what these sessions are designed to do is just to provide you with a broad overview. And then in the next session, we'll actually have a hands-on session where we will run multiple analyses together. So before that time, you might play around with a pipeline and really see how it works. But then if you have any questions or you want to understand how to find the data, upload the data, put it into the platform, and then start comparing the different sequences, we will do that together during the session. And we will focus this session on full genomes, so whole genomes that have already been assembled. After that, after you will have some time to play around with some of those pipelines and reproduce some of the analysis on your own or try an analysis of your own, we will talk about how do we analyze the pipeline results. So once we got a result from a pipeline, what do we do next? What types of different file outputs will we be looking at? And how do we actually understand the significance of what we found, right? And we'll discuss that in the context of a Q&A session with some practical examples. Then we'll start talking about transmission and infection spread. How do we understand the relationship between genomic data that we look at, so just from the standpoint of a genome, and the process of infection that takes place in a single host, in a single patient, for example, and then all of a sudden spreads to many. And so how will we look at viral adaptation and transmission in the context of the biology of virus and the replication that it goes through? We'll then talk about the disease progression and outcomes. So what are some of the important features that characterize differences in the ability of the virus to enter the cell, replicate within the cell, eventually causing the host to either be very sick, and how does that contribute to the spread of the virus, or potentially die, and why is that maybe a bad thing for the virus as well. And so really we'll talk about that evolutionary relationship between the host and the pathogen, where one needs to keep replicating using the host resources, and the other one needs to survive and suppress that replication, but both actually change as a result of that interaction. So we'll talk about that virus host circuitry, or virus host interaction. Then we'll talk about a hands-on session. So practically speaking, what can we learn from available data, right? So a lot of the discussions are going to be around, okay, what has been published, what has been known, what kinds of issues are highlighted, but then we need to bring it down to a real data set, right? And so we'll take a few data sets here, and we'll talk about the origins, and we'll talk about a pandemic as opposed to an epidemic, as opposed to just a single infection. How do we characterize some of those things by just looking at data? Then we'll talk about the rate of mutation. So we'll talk about different types of mutations and different ways to characterize mutations. Some of them based on knowledge, some of them based on, for example, what we know mutation can cause as a change in a codon and the resulting amino acid or residue that comes out of that. But also we'll talk about how can we understand fitness, and how can we understand random as opposed to accumulating beneficial or detrimental mutations, and what kinds of data sets do we have to characterize that kind of difference in data. We'll also talk about how that type of understanding can lead to effective strategies in, for example, vaccine design, or association between the spread and the danger and the effectivity of a specific strain, and some of the concerns, public health concerns, or intervention concerns that people have. And so we'll talk about some of the protein structure function. We'll talk about some of the examples where you can see here an important mutation that was used for understanding the different strains that were more or less effective. How can we study and generate that kind of data, and how do we analyze that kind of data and interpret what it actually means? And then we'll talk about host pathogen interaction. So we do have omics data from cellular response or the host response. We do have the genomic variation that we can observe. How do we integrate that data together, and what can we learn about the process of integration and some of the challenges that come out of there, right? So, and two main strategies, the strategy of knowledge driven and the strategy of kind of unsupervised or data driven. So we'll talk about that. And then right after that, what we feel by that point is that you're equipped for doing your own project, and you can start working on a project either before that and still participate in the sessions that we hold to ask questions and get support, and of course share your results if you're interested. But also after we complete the sessions, if you want to work with someone else in the program and collaborate on a project, and that would be a way to leverage some of the different skill sets that people have. Some have more computational skills, some have more biological experience, and some might be able to find an interesting data set and be able to extract all the right information from the databases that we will discuss. So we'll talk about this in the context of this session, and really we hope to help you plan the full analysis from beginning to the end, and also introduce you to ways of effectively finding the data set and bringing the data set onto the analysis, into the analysis plan, so that you are doing that effectively. So that is a session, but also a whole month that you will have to work on your independent project. To get a sense of what this these sessions might look like, we recommend that you visit our YouTube channel and look at some of the recent recent video recordings of those sessions that we held at different universities, including Louisiana State University and Loyola University, Georgetown University, and others, where you can see the interaction with the students, and some of the questions that are being posed, some of the data sets that we review. All of that is in the context of COVID-19. But you will see how that applies to some of the other data sets that we will discuss. Again, what are those projects that we started preparing? So we will talk about tuberculosis. We'll talk about the polio and other enteroviruses. We'll talk about enterovirus D68 and acute flaccid myelitis in kids. And we'll talk about Ebola. But also we'll dedicate time to speaking about SARS and COVID-19. In the process, any of you can propose a project that might be interesting to you, and we hope to contribute to the analysis that you'll do. So in the context of each project, we'll actually take a detailed look at the meta data that we have available for the omics data that we have. So here you can see, for example, the spread of Ebola in West Africa. And that is an important component of the genomic data that we have access to, right? And so in the context of that same Ebola virus project, we'll review some of the important articles and publications that came out during the time of that early 2014-2015 outbreak, and eventually how the understanding of what the virus does and how it interacts with the cell, we can identify specific critical proteins, and how those proteins over time led to the development of a vaccine, and what are some of the alternative vaccine designs that have been proposed throughout that process, as well as how do we understand the immune response and the challenge of sustained immunity from vaccination. We'll also talk about the interaction between host and the virus. So again, a lot of this is going to be focused on finding the right data. So we'll go and we'll find the data together. We'll take some extraction from the full data set that is available to prepare small exercises that you can work through fairly quickly, in the hopes that you will be able to go back to the full data set and find data that is interesting to you and develop something novel. So we'll talk about how to navigate some of these repositories and how to make sure that you select for the right combination of elements to make a good data set for comparison. And when we talk about these smaller examples, we'll actually try to focus on some reproducible analyses that will have clear-cut outcomes for you to be able to make sure that you're doing the right thing and you're actually getting the result that is expected from an analysis, right? So for example, as I mentioned before, how do we compare different strains of coronaviruses and understand which one is more related to which one and how are they different? And then map that kind of difference on some important element, for example, for vaccine design, like the spike glycoprotein, where we see a difference between different areas with abundant mutations, and how do we interpret what is the function? And we'll try to link these different levels of information that we are processing and we are looking at to be able to make more interpretable results from the analysis by using different strategies for comparison and different types of input data that will lead to different types of outcomes. So again, I hope that the questions about access to the different resources, the questions about different sessions that we will have in the schedule and how long you will have access to different things have been answered during this session. If you have a question about, what are these sessions going to look like and how am I going to be able to use the tools, I highly recommend, again, going back to our YouTube channel and seeing some of the previous recordings. And if you have any more specific questions, I'm happy to answer those now. And thank you, Bipsha. Yeah, so now in chat, you can see the link to our YouTube channel where you can see some of the previous sessions. All right, great. So if there are no more questions, we're going to start on May 4th. That's going to be Monday next week. Okay, there is a question, sorry. How can I get a mentor to start a specific project? Okay, great. So, okay, first about a mentor. So we will have a group of people, 20 to 30, maybe 50 people that will participate during different sessions, and we will introduce some of the experts throughout those sessions. So we have some experts on molecular biology of different viruses that will join some overview sessions and be able to answer some questions. We will have some bioinformatics experts that will be able to talk about the analysis methods and help you with that. You do have an opportunity to interact with our team throughout the session and communicate with us. We will have a forum dedicated to this program where all of those questions will be answered either by us or by somebody that can answer that question, and hopefully find someone that is going to be a good match for your project. Okay, Martin Blazek. If I registered for the course at the EDU site, should I be able to log into the server site? Yes, you should get an email before Monday on how to access your account on the server. And if you have not, then over the weekend, I'm sure you will. But make sure that you make those, you pay attention to those emails, because they will come from by email, and you will have a different login from the EDU site. Will there be an opportunity for publications with ResearchGate? What is the interface with R and Python and Java and SQL? So first, publication. So obviously the publication is something that you can do in several ways. Typically, you might prepare a poster and maybe submit it to a conference. You might do a full publication on some meta-analysis, right? Or if you have access to some novel data. So I think that's a little bit, of a question to you, whether you have the right data set and you can develop the right analysis and complete it during the time of this program. We hope that you do. If not, I think you can continue working on your project even after this program ends. We will obviously share with you all the data if you want to close your account, or if you would like to keep using the account. There are ways to transfer that data to a research account. The interface with R and Python and Java and SQL. So we will show you how to translate the analysis that you have done on our platform to R. For some of the analysis, if you want to do that analysis in R, the processing, for example, does not make a lot of sense just because of the size of the data and the pipeline intensity on the computational side. You typically would use a server to doing something like that. So if you have access to that server, it's probably something that you can match, right? So you can find the methods that were used in the analysis that you did here and map it over to some other environment where you would like to work. For some of the post-processing steps, right? So once you've generated a table, let's say, how do you analyze that data with R or Python? And whether you can organize your data in some kind of a database so that you can apply SQL, right, for searching maybe for the right types of data sets, etc. So we'll touch on that in some general terms. This program is not focused on helping you learn how to code or use databases. We will more apply those skills from a conceptual standpoint, because I think there's a lot of user-friendly tools that make this possible in a short time frame, right? For the longer time frame, we can actually discuss that and point you to the right resources. About making payments, again, maybe Bipsha, you could help Jamila to finalize her registration. So again, if any one of you needs some help with completing the registration, I know it could be a little bit complicated because we take the time to review each submission. Please email Bibsha directly, or she should be available on chat. So in fact, I'm just going to share that with you really quickly. So the process of registration is going to start from this page, which is a form where you register right here. And once you submit this, you will get an email notification on how to register on the educational portal that we have, which is where you would complete that registration. But Bibsha will be able to answer your questions there on chat now and take your details if you need more information. Databases, yes, excellent question. Let me point you to actually a collection of different resources that we have prepared partly for this program, but in general as well. So this is in a blog post that Mohit Mazumdar prepared just recently, right here. It has a collection of different resources and databases that you can start navigating. Thank you, Lisa. Okay, yes, so here you can see a whole collection of different either visual and educational resources that you can learn about some of the viruses that we'll discuss. You will find here NCBI and different resources and what kinds of data you can find there. NCBI virus, for example, is a dedicated resource where you can find all of this. A resource where you can register for free, JIS Aid, which was originally designed for influenza research, but you can actually find a lot more there, including COVID-19 and other types of viral pandemics or epidemics. Where do you find some gene expression data sets on NCBI Geo? Viper is a great resource, originally again designed for influenza research database, but now a lot of different virus tools are available there. Our platform, obviously, and some of the different sections that we discussed here and how to use those. And then also some of the visualization data where you can also download data, for example, NextString. Okay. Another question, we expect to work on our project further. Does that include any payment? So after the program is going to be over at the end of June, you will be able to make a decision, right? If you want to continue and work on the platform, you are welcome to get a research license. If you would like to continue working on it in terms of, for example, writing your publication or visualizing data, you might not need the platform. So feel free to decide for yourself exactly how you want to manage that. Just to give you a sense of the platform and what would be available after this program is over. We do have research licenses or educational licenses if you're still learning. We do offer support for all of that as well, but we will also have some other programs designed specifically for those interested in research. So Mohit, maybe you can share some of those links for the upcoming programs if you have that, where it's not sessions like this, informational sessions, it's more sessions dedicated to analysis and discussion of analysis results. Thank you. Okay, so again, thank you everyone for joining today. This session was recorded and we will post the recording online. If you would like to see that recording, we will email it to you, but also it will be on our channel, on our YouTube channel. So please subscribe to the YouTube channel and see this recording and some of the other previous workshops that we did. as well as some of the other programs to get a sense of what that how that works. How can I get a mentor to start a specific project? So depends on what kind of mentor are you looking for. I'd be happy to communicate with you directly if you want. So please send an email to Bibsha, who handles those kinds of questions, and we can take it from there, because we need to learn more about your project and your interest so that we can match you with the right person. All right, great. Thank you everyone again for joining. We would be meeting on Monday for those of you that have already registered, and looking forward to start learning about you and your interests as well as you submit your forms. If you have not done any bioinformatics, that does not mean that you cannot join this program or some of the other programs that we have. And we welcome you to start by going through our Introduction to Bioinformatics course, where you can learn about the impact that bioinformatics is having on life sciences. All right, great. Thank you, Rare Seas, and looking forward to seeing you as well. Take care, everyone, and have a great weekend. Thank you, Chandrani and Martin. See you guys on Monday.